Today we discuss the new topic, that is population genetics, which is related of a new syllabus of NTA. So first we discuss about what is population genetics. So we clearly know the content of genetics which is given by the scientist Georg John Mendel, but here we completely discuss about the frequency distributions. A calculative methods or a calculation which is properly given as a form of what? Frequency distributions. So to study about the frequency distributions or genetic frequency is taken as what? Population genetics. To calculation of what? Frequency distributions of a population is called what? Genetic populations or the population genetics. So the frequency distributions or the population genetics which is depend upon 3 points value. One is gene pool, second is gene flow, and third one is gene frequency or frequency of genes. First comes to gene pool. So what is gene pool? The total number of genes present in a individuals. The total number of genes present in a individual of what? Gametes. Then it is called what? Gene pools. So that individual which is from where? Of one populations. So the total number of genes which is present in the gametes of a population is called what? Gene pool. Now after that one, what is gene flow? The transformation of the genes from one population to another populations. एक population से दूसरा population को अगर क्या transfer हो गया, gene अगर transfer हो गया, तो उसको मैं बोलूँगा क्या? Gene flow. Now comes to what is gene frequency. So you clearly know allelic difference. You know. So the frequency of allelic difference, difference of a gene in a population. So what is gene frequency? The alternate distribution of frequency of gene in a L. Frequency of gene in a L. Otherwise we can say that the difference of the distance of LL, the distance of LLs of a gene in a population is called what? Gene frequency. Let's we take one example. So the example is, in a population there is a two blood group is observed, that is M and N. So in a population of gene frequency distributions, once we discuss about one example characteristics and what? There is a two blood group is observed, that is what? M and N. So दो टाइप का क्या दिख रहा है? ब्लड ग्रुप दिख रहा है. सो Mn जो ब्लड ग्रुप है वो कितने इंडिविजुअल का दिया है ना? टोटल नंबर ऑफ पॉपुलेशन व्हिच इज गिवन 100 नंबर ऑफ इंडिविजुअल. हाउ मेनी नंबर ऑफ इंडिविजुअल? 100 नंबर ऑफ individuals. Clear? From that 100 number of individuals, the contents of genes, MM gene which is given as a form of what? 50. And the characteristics of M and N, the hybrid characteristics is given as a form of what? 20. Are you getting my points? And NN blood group of genes which is as a number of 30. So what is our calculation? The calculation here, the point is what is the frequency of M and N gene? What is the frequency of what? M and N gene. So how we calculate? Here given the data is MM is 50, NN is 20 and NN is 30. So our target is what? What is the frequency of M and N? So we calculate the frequency of M and N. So for the calculation of frequency M and N, we calculate that. We clearly know the gene of M. How we calculate? We calculate that gene of M first. Gene of M is equal to m is equal to 2 into m plus, what is the hybrid is already given, mn 20 plus mn. Are you getting my point? So 2 into 50 plus mn how much? 20. So 120 is what? Gene of m gene. Okay. Now comes to gene of n or n gene. 2 into m plus mn. The meaning is what? 2 into 30 plus mn how much? 20. So complete is how much? 80. So total number of gene, how much you got? 200. तो हमें जीन फ्रीक्वेंसी लेना है, किसके बारे में दिया है? एक ब्लड ग्रुप एक बारे में दिया है, उसमें एम और एन ब्लड ग्रुप का दोनों है, जिसमें एम ब्लड ग्रुप वाले कितने थे? 50 थे। एन ब्लड ग्रुप वाले कितने थे? 30 थे। एम वाले मिक्स्ड ब्लड कितने थे? 20 थे। तो हमें निकालना क्या है? जीन फ्रीक्वेंसी निकालना है। तो जीन फ्रीक्वेंसी के लिए मैं क्या निकाला? जीन ऑफ एम कितना है देख लो, जीन ऑफ एन कितना है देख लो, तो मुझे क्या मिल गया? टोटल नंबर ऑफ जीन मिल गया। तो आपको मिला अब क्या? टोटल नंबर ऑफ जीन मिल गया। अब मैं देखूंगा कि फ्रीक्वेंसी क्या है उसका। सो फ्रीक्वेंसी ऑफ जीन, नाउ टारगेट फॉर व्हाट? फ्रीक्वेंसी ऑफ जीन। सो फ्रीक्वेंसी ऑफ एम जीन, फ्रीक्वेंसी ऑफ व्हाट? एम जीन इज p. M gene is what? P. So the calculation is m by m plus n. M by m plus n. M how much? Already we know. Just we came, sir. Just that is came 120 gene m. M plus n already 200 total number. So that much how much it you got? 12 by 20. The meaning is what? 2 6 2 10 0.6. Okay. Now the frequency of n gene is q. So q of frequency, that is what? N by m plus n. N the meaning is completely calculation is 80 by m plus n is 200. 1 0 cut, 2 4 2 10 0.4. So what we got? We completely distribute the frequency. The question is what? The question is what? Frequency of m gene and frequency of n gene. So frequency of n gene we got what? 0.6, and frequency of n gene we got q, that is what? 0.4. So the calculation one value is came, that is p plus q is equal to what? 1. P plus q is equal to 1. The meaning is over there na 0.6 plus 0.4 is equal to 1. So what you got? P plus q is equal to 1. Once this is one type of question. Once any other type of question, once it is came. How? Here given completely in a complete population of ecology, in a completely populations, in a normal genetics of population or population of genetics, there is given 0.7 number of individual, 0.7 number of individuals are frequency of one dominant characteristics. Find out the recessive content or other dominant characters, then how you calculate? So P plus Q is equal to 1. P is always calculated as a form of what? Dominant category. Q is calculated as a form of recessive contents. So P is given 0.7. So Q is equal to how much? 1 minus P. The meaning is 1 minus 0.7 is equal to 0.3. Then you can able to calculate what? Q. Later the scientist who had wing. So head and wings, head Weinberg. So Weinberg is the other scientist. Head means head is the other scientist. Two scientists together, formation of a calculation equations or calculative of what? Equation of what? Frequency distribution of a alternative generations. Which generation? Alternative generation. He gave a completely explanation about the frequency distribution which can able to calculate as a form of what? P square plus Q square plus 2PQ is equal to 1, where P square is always dominant of what? Dominant of what? Homozygous. Homozygous. Dominant of what? Homozygous. Same as Q square is recessive. Homozygous. And PQ is what? Hybrid. Are you getting my point? So the calculation is P square plus Q square plus 2PQ is equal to 1. Clear? So now comes to population genetics Hadwin principles. Hadwin the principles is P square plus Q square plus 2PQ is equal to 1. Once we solve a problem on the base of P square plus Q square plus 2PQ is equal to 1, Then, so here given the data, the populations, in a populations, the homozygous recessive AA, what? Homozygous recessive AA is 36%. Then find out the heterozygous, find out the frequency of find out the frequency of heterozygous genes. Heterozygous genes. Are you getting my points? Right? So here only a data given that one, that homozygous recessive is 36%. So how we calculate? The calculation is we properly know that homozygous recessive, the meaning is what? Small a small a. Small a is equal, small a, the contents of homozygous recessive small a is q square, recessive content is q square, yes or no? So q square is equal to how much given? 36%. So q square is equal to 36 by 100. Okay. Then q is equal to how much? Root over 36 by 100. The meaning is 6 by 10, that is 0.6. How much? 0.6. So we already got q is equal to 0.6. We already got what? Q is equal to 0.6. Now we clearly know the formula P plus Q is equal to 1. Okay. So given what? Q is equal to 0.6 already we got. Then P is equal to 1 minus Q. So P is equal to 1 minus 0.6 is equal to 0.4. So what you got? You got that the value of frequency of P and the value of frequency of Q. Then the calculation of what? Here already given that, given that what? The find out the characteristic of heterozygous. So on the basis of formula you clearly know P square plus Q square plus 2PQ is equal to 1. Where the 2PQ is what? Heterozygous. 2PQ is what? Heterozygous. Then the value of 2PQ, then what? 2 into P how much given? 0.4 we got. And Q how much we got? 0.6. 6 4 ja 24. 2 jar kitna? 48. So how much the frequency distributions of 2PQ? 48%. Are you getting my points? How much it is? 48%. Let's another question is once we solve a African population, in African populations where there are 9% of peoples are affected with the hemophilia. How many percent of peoples are affected with the hemophilia? 9%. You clearly know a hemophilia contents of disease which is mostly recessive genes is highly affected. Which gene is affected? Recessive gene is highly affected. So once it is a recessive gene is highly affected, then the meaning is what? It is already given Q square is equal to 9%. Right? Then Q is equal to how much you got? 0.3. Yes or no? Okay. So once you got Q is equal to 0.3, then P is equal to how much? 0.7. Because we properly know P plus Q is equal to 1, then P is equal to how much? 0.7. So here question is given, so question is what? A African, in Africa or African peoples where 9% are homozygous recessive. So 9% are which content? Homozygous recessive. Once 9% is homozygous recessive, then what we find out? Then mixed of heterozygous. So heterozygous again 2 of PQ, 2PQ characteristics. 2 into 0.7 into 0.3. 7 3s are 21, 2s are how much? 42. So 42% of the people who are heterozygous. 42% of the people are as a form of what? Heterozygous. Are you getting my point?